Many of those repair and resiliency reactions depend upon NAD and all of the accumulated insults involve, losses to the NAD system. So I think there is a use case for nicotinamide riboside in human health. It may be, in fact, more important as we age. There's lots of clinical trials involving NR to test these hypotheses. So I think aging is
So hello and welcome to the Sheekey Science Show, where in this video I had the opportunity of speaking with Charles Brenner, a professor at the Department of Diabetes and Cancer Metabolism at the Beckman Research Institute of the City of Hope National Medical Center. He has contributed to the field of NAD metabolism and is passionate about demystifying aging research. I hope you enjoy our conversation. So hi Charles, thank you for joining me today and welcome to the Sheekey Science Show.
Great to meet you. Thanks, and it's nice to be here.
Great. So, I brought you onto the channel today to talk about your research and your knowledge, in particular about NAD metabolism. And so I thought we could begin there. And I know it's quite a big question, but if you're able to somehow summarize what is NAD metabolism and why is it important in the body?
Great. Thank you. It's a great opportunity to, and you're going to hear some noise in the background here, unfortunately, but NAD refers to 4 coenzymes that are the central catalysts of metabolism. And so I like to think of metabolism as the collection of all the reactions that convert everything that we eat into everything that we are and everything that we do. So it's not just converting food into ATP biological energy, but it's converting food into biosynthetic intermediates in order to make everything in our cells allow us to replicate our DNA, make RNA and protein, make lipids, and repair all of the molecules in our body. So all of that is metabolism. And not in every single metabolic step, but within 2 or 3 steps of every metabolic step, there's movement of high energy electrons. And so the source of the high energy electrons for life on this planet is the sun. And NAD in the oxidized form, NAD+, captures electrons in processes like glucose oxidation, amino acid oxidation, and lipid oxidation, forming NADH. And then NADH is a hydride or an electron donor to a number of reactions. NADH contributes lots of electrons at the intermitochondrial membrane to generate ATP by oxidative phosphorylation, but the equally important components of metabolism are biosynthetic, and that is mostly done by a pair of coenzymes called NADP and NADPH. They're made from NAD, and they're essential for making all the nucleotides in your body. They're essential for making all the lipids in your body and for detoxifying reactive oxygen species. So that's how central NAD metabolism is to life on this planet.
Yeah, so I mean, as you explain, it's involved in so many different processes. And so it kind of raises the question as, how do you go about studying these different metabolites?
Well, I grew up as a biochemist, and 20 years ago, I was really interested in a particular enzyme called glutamine-dependent NAD synthetase. And I won't go into why I thought it was interesting, but it has 2 active sites. One of them takes glutamine to glutamate and mobilizes an ammonium, of all things. And the other is called an NAD synthetase domain that uses that ammonium group to perform what was then considered the last step in NAD synthesis, from tryptophan, from nicotinic acid, and in yeast also from nicotinamide. And I thought that enzyme was interesting because it had two active sites and it had to coordinate those reactions, and it is required to make NAD, which is the central catalyst of metabolism. And so, there's a body of work to be done on that. But when I started learning more about NAD, I thought that there were potentially unsolved problems in NAD metabolism. And I questioned, in fact, whether tryptophan, nicotinic acid, and nicotinamide were the only ways of making NAD. And I thought that there might be a nicotinamide riboside pathway to NAD, and that use of a nicotinamide riboside as a NAD precursor in yeast and potentially in humans would potentially reveal another pathway to NAD synthesis. And so I looked for unsolved problems in NAD, and I continue to find lots and lots of things to work on. And probably the most important giant category of things that we're interested in is the regulation of the NAD metabolome, because you would think, I used to use the terms the crown jewels of metabolism. I know it's a week of mourning in the United Kingdom right now, but crown jewels are locked inside of a safe and put inside a vault, and the vault is patrolled by armed guards, and there might be a moat around a castle, right? And so crown jewels are very, very protected. These are the central catalysts of metabolism. But it turns out that NAD coenzymes, despite their crucially important roles, are exposed to what I call conditions of metabolic stress. The NAD metabolome is not stable to conditions of metabolic stress, including DNA damage, oxidative stress, and then I could name you a dozen physiological challenges. And so we're really interested in how the NAD system gets disturbed, and then how it tries to repair itself, and then how these things get altered in diseasing conditions, and then what we can do to improve the human condition, understanding all of this.
Yeah, so you brought up many different points there. And so one of them you've mentioned is nicotinamide ribosides. And so it seems to me you mentioned how you deduced that there were these unanswered questions in metabolism. But how did you get about that deduction? Was that you looked at the biochemistry and inferred that maybe you thought there was some mechanism that would take you from NR to NAD, or how did you get about this?
Yeah, essentially. So there were, 3 or 4 different lines of reasoning. One is that I grew up as the intellectual grandson of Arthur Kornberg. My graduate advisor, Roberta Fuller, was Arthur's graduate student, and Roberta and I were working in Arthur's department of biochemistry at Stanford. And kind of the legacy of Kornbergian biochemistry goes all the way back before he worked on DNA replication. He worked on NAD synthesis. And Arthur purified an enzyme, I think first from a bag of potatoes that he bought in Maryland when he was working at NIH. He purified an enzyme from potato that interconverted NMN and NAD. So now we call it NMN adenylyl transferase. Old school biochemists will study a reaction in either direction that's more convenient to them. So Arthur, had NAD available to him, So he actually characterized this reaction in terms of it splitting NAD into NMN plus ATP, I think, because the reaction actually goes the other way, right? ATP and NMN form NAD and AMP pyrophosphate. So, so I knew that there was an enzyme that would convert NMN to NAD. I knew that from biochemical activity that was described in 1950 or maybe even earlier than that. And when I looked at the yeast genome, I was a yeast person at the time. When I looked at the yeast genome, there wasn't any gene that was annotated as NMN adenylyl transferase. This is getting a little technical. I should have a whiteboard here, but essentially there were a bunch of gene names there that were assigned to do particular things. There was genes called NMA1, NMA2, and they were assigned as having the activity of taking NAMN to NAAD. Then my enzyme, glutamine-dependent NAD synthetase, was going to convert NAAD to NAD. So, as you know, would be writing this on the whiteboard. It would be all of these reactions are going up to NAD from the left side, but NMN adenylyl transferase would be making NAD from the right side. But there has to be something that makes NMN.
I see, yeah.
And well, I know there's an enzyme that will convert NMN to NAD, but no one's described an enzyme that makes NMN. Okay, so that's one piece of information. Then there's another thing. There's a failed cancer drug. You read widely, right? So there's a failed cancer drug. It's called thiazofurin. There's another sort of loser of a cancer drug called benzamide riboside. You might even come across benzamide riboside in the PARP literature. Both thiazofurin and benzamide riboside are basically analogs of NR. They've got a base that looks like a nicotinamide. Benzamide is exactly nicotinamide, except it's a benzene ring rather than a pyridine ring. Nicotinamide and nicotinic acid have a nitrogen in a six-membered ring, right? Benzene's got all 6 carbons. So, and riboside, meaning it's got a ribose linked to the base. And benzamide riboside thiazofurin, which is a sulfur-containing nicotinamide analog linked to ribose. Both of these, not very effective cancer drugs, are cytotoxic because they form NAD analogs. Well, isn't that interesting? You have something that you can draw on paper that looks like nicotinamide riboside, and it's super toxic. And why is it toxic? Because it's making an NAD analog. Therefore, there must be a nicotinamide riboside kinase, right? And by the way, vertebrates have an enzyme called NAMPT that takes nicotinamide to NMN.
Mm-hmm, yeah.
So vertebrates certainly use something similar to the potato enzyme that takes NMN to NAD. And no one's described a nicotinamide riboside kinase in vertebrates. Now, nicotinamide riboside is a molecule. It's been named. I didn't come up with the chemical name for it. It even had an older name than that. It was called factor V. You love it when the old microbiologists have a name for a factor. You know what factor V came from?
No, I don't. I mean, there's I've had it in the protein context, but not as a metabolite.
So factor V. Oh no! It's not blood coagulation factor.
Yeah.
No, that's a good question. That's a good guess. but it's nothing related to that. So it actually comes from not a flu virus, but a flu-causing bacterium called Haemophilus influenza. Haemophilus influenza. And now only 6 crazy microbiologists in the world want to grow Haemophilus influenza, right? Like, who would do that? And why do you think, and maybe when this is on YouTube, you can pop up a question and have an answer box or something. Why do you think they named this bacterium Haemophilus?
So, well, I guess something to do with blood, but
it loves blood, right? So it actually needs blood to grow this bug, right? So it means you can't grow it on LB medium. You can't grow it on minimal bacterial medium, M9. You need M9 salts or whatever, and then you need some blood, right? So that's where microbiologists turn into chemists and biochemists, is they have to fractionate blood, and they have to see what is it in blood that Haemophilus influenza can't make on its own. Turns out it's 2 things. Haemophilus influenza has a very small genome. It was actually one of the first bacterial genomes that was sequenced in I'm not sure you were born. Literally, I think might have been one of the first 1 or 2 or 3 bacteria sequenced, and Hamilton Smith and others. And it does not have a heme biosynthetic pathway, so you have to give it heme. And it can't make NAD from any normal thing. Like, it can't make NAD from tryptophan, it can't make NAD from aspartic acid, which most bacteria can, and it can't make NAD from nicotinamide, and it can't make NAD from nicotinic acid. Okay, so initially what had factor V? Factor V. What initially had factor V activity was NAD itself, and then turns out NMN also had factor V activity, and then the smallest piece of NAD that had factor V activity is NR.
Ah, okay.
So it turns out that Haemophilus influenza will take NR, and then it has a nicotinamide riboside kinase and NMN adenyl transferase to make. So it has a super tiny pathway, 2 enzymes, to take the nucleoside precursor of NAD to the mononucleotide to the dinucleotide. That's the only thing in the H. influenza genome. And so NR was in the literature as an NAD precursor for this really whack bacterium. And I thought, well, yeah, but no one's ever tried to see whether it could be a precursor in a eukaryote. And I have a system now because I'm mutating a glutamine-dependent NAD synthetase. And if this works the way I think it's going to work, it's going to bypass the essentiality of NAD synthetase. Because NAD, I told you, there's a yeast genome database by now with assumed to be correct metabolic diagrams. I'm the one that thinks everything's wrong, right? So I don't accept the diagram as being full and correct, and neither do I really accept the idea that NAD synthetase is going to be essential under all circumstances. And basically, we did an experiment to see whether you can bypass the essentiality of that gene with NR, and you could. So NR allowed you to live without NAD synthetase, which meant there's got to be another way, because NAD synthetase is required in yeast from tryptophan, from nicotinic acid, and from nicotinamide. doesn't have NAMT. And so that was an awesome insight. And then we said, now we have the way to clone the NR kinase, because now if we find the real NR kinase, then that's going to block the ability of NR to bypass the other pathway. So you probably have two kinds of listeners: the super nerdy science listeners who are grooving with all of this, and the other ones that just want to improve their health and stuff like that, and they're like, what is he talking about? But anyway, that's back almost 20 years ago. That's how I got into NAD. That's how we found the nicotinamide riboside kinase pathway. And then, yeah, we always, like, people tell us stuff, or we see it in books, and we just say, what's the evidence for that? And usually we find it's not entirely correct, and then we find a way to figure out what is correct.
Yeah. So, I mean, I think I followed myself about 75% of that. So I think I have to
Cool.
that's
Great.
somewhere there. But I think you raised a really interesting point, which is about experimental technique and method in terms of actually deciphering these different pathways. And I guess, I believe in terms of how you go about quantifying the levels of these different metabolites, you use like mass spectrometry. But I guess even in that, there's limitations. So would you be able to elaborate on what you think are like the gold standard ways of actually being able to quantify these different metabolites?
Our method is the gold standard.
Okay.
So you need internally standardized, quantitative, targeted NAD metabolomics, and that was developed in my lab. Our first publication on it was 2013 with Sam Trammell, who is now, I think, on the faculty in Copenhagen. And basically, it's a really cool method. We get yeast to make a C13 version of the NAD metabolome, and then we quantify that yeast extract, and then we dope that yeast extract into any sample that we measure, and then we can put NAD metabolites on a picomole, on a molar scale. And that's really important, because somebody may say, oh, there's like a twofold difference in some metabolite, but if it's, 2 micromolar versus 1 micromolar, it's almost certainly not going to be interesting. Somebody is calling me over and over again. Let's do a little timeout. So we do targeted quantitative NAD metabolomes. We're actually expanding our method to cover many more metabolites.
Interesting. And so I think that another thing, is it mainly looking at blood samples, or like you work with different tissue samples? And in terms of that, can you also fractionate the samples prior to your methods to see where within the cell these metabolites are?
So there's a saying, the difficult we do immediately, the impossible takes a little longer. You can do any tissue that you want. Right now we're doing something in mice that's liver, kidney, and blood. We've done human liver from people with alcoholic liver disease, done lots of heart metabolomics, lots of blood metabolomics, brain, whatever, CSF, anything that you want to do. We generally look at whole blood rather. We've some publications where we made PBMCs, but we think that time is of the essence. And with that, what brings up the exact question of subfractionation. So sure, you can subfractionate, but then what are the controls that you're going to do that will be sufficient to say that your, let's say, mitochondria are so intact that you are really looking at a mitochondrial NAD metabolome as opposed to what's left after you pelleted your mitochondria. So you need complementary methods. There's now things that you can do with fluorescence microscopy that are reporter groups and stuff like that. So we like to collaborate and use other methods as well.
I see. And so, I mean, there are limitations with trying to quantify it, but it still gives a kind of reliable readout in terms of you see, I believe, declines in NAD plus, for example, in response to these different metabolic stressors such as diabetes, etc., as you've mentioned.
Yeah. So we've done experiments with high fat diet in mice and then induction of type 2 diabetes from there. And you see a disturbed NAD system in the liver. We've done experiments in peripheral neuropathy, central brain damage. Others have done noise-induced hearing loss. I think some folks are now doing age-induced hearing loss, where there's a depression of cochlear NAD. Certainly DNA damage induces pretty dramatic changes in the NAD system with PARP activation, and then PARG activity breaks up the PAR polymers. And so we're interested in all of those dynamic changes in the NAD system.
And currently, I mean, do you think are the main reasons for this decline in NAD+? You've mentioned PARP enzymes, which kind of consume NAD+ to perform their reactions, but are there other explanations that might account for the decline?
Yeah, well, and the first thing is that there's a lot of articles in the review literature that say, NAD declines in aging, and I'm always saying, okay, who measured that, and in what tissue, and using what method? And so a lot of it has been storified, and we don't really see that blood NAD, necessarily declines in aging populations. The evidence for liver NAD decline is much better, but I think it may be driven by inflammatory processes. And so I always question whether changes in the NAD system are episodic and due to specific insults, or whether they're strictly time dependent. And I don't think that it's all that well established. So I always step back and, try to talk about what the actual data say. Now, one of the really striking cases in which NAD comes under attack in a disease process is in heart failure. And it's a strong interest to us because in multiple models of heart failure, NAD comes under decline, and then the nicotinamide riboside kinase 2 gene spikes up. And we're trying to determine whether it's a low ATP state, which there's some evidence for that, turns on the nicotinamide riboside kinase 2 promoter to try to kind of homeostatically, restore NAD balance, or something else. But there are a number of enzymes that can increase NAD consumption. So when PARP1 and PARP2 get activated, and that's usually due to DNA damage, then NAD can be kind of degraded and turned into the ADP ribose polymer. In infection and inflammatory conditions, we've shown that the interferon system turns on 5 different members of the PARP superfamily. They're mono ADP ribosylating enzymes. They're turned on transcriptionally, and then they degrade the NAD. Seems like it's a response to an innate immune response to viral infection. And so we're actually trying to figure out what are the cellular and or viral proteins that are morelated when there's double-stranded RNA in a cell and when there's virus in a cell. There's an enzyme called CD38, which has multiple membrane orientations, some outside the cell, some inside the cell, and some inside the lumen of the endolysosome. And that can convert NAD to cyclic ADP ribose that can then mobilize calcium and lead to a number of really interesting changes in signaling and cell mobility. And then there's an enzyme called SARM1, and SARM1 basically gets kind of angry when it sees NMN. And so we were talking earlier about NMN adenylyl transferase. NMN adenylyl transferase in a neuron is located on the outside of the Golgi apparatus. So the peripheral neurons are the longest cells in your body, right? So from your sciatic nerve to the tips of your fingers and the tips of your toes. And that's a very, very long axon. And so that cytoplasm and the axonoplasm, if you'll allow me to partially coin a term, I think it's a real word, that axonoplasm is dependent upon the Golgi localized NMN-AT2. And so it turns out that when fast axonal transport is disrupted and when an axon is damaged, NMN-AT2 basically is an unstable protein. This is the work of Michael Coleman in the UK and collaborators. And NMN accumulates, and then NMN activates an enzyme called SARM1, and then SARM1 can turn NAD into cyclic ADP ribose and initiate Wallerian degeneration. So it's actually very funny because there are people that we've shown, and many other people have shown, that NMN, which some people, for reasons I don't understand, I think it has to do with influencers, NMN is converted extracellularly to NR, and then NR goes into cells and repletes the NAD metabolome. But there are people that think that NMN magically gets into cells or gets into cells through some transport mechanism. But it's actually been shown that when you push NMN into cells, it activates SARM1, and that leads to Wallerian degeneration. So there's just a lot of messes in the NAD literature that I'm involved in kind of cleaning up.
So before we come back to some of you've mentioned just a load of different topics there, and some of them will come back to you, but just to start with where you ended, you mentioned earlier, like NR can get converted to NMN. So how does that play into, if you're increasing NR levels through supplementation, does that also somehow activate someone because it gets converted to NMN?
I don't think so. NR gets into cells, nicotinamide riboside kinase turns it into NMN. NMN adenylyl transferase will convert the NMN into NAD. Then there's NAD, NADH, hydride transfer. Some of the NAD gets converted to NADP, and then there's the NADP, NADPH redox couple. So it's really only in cells that are have a problem with NMN-AT activity, where NMN will accumulate. Unless, if you had some liposome delivery or you're transfecting in, bolus amounts of NMN, then you could have an NMN problem. And that, actually, it's been demonstrated. There are NMN analogs that are cell permeable. Unlike NMN itself, there are NMN analogs that are cell permeable. And those analogs, because they're cell permeable and it's dose dependent, obviously. But if they're not great substrates for NMN adenyltransferase and they're cell permeable, then you could have this alarming, signal in a cell that there's a lot of NMN there. But I think generally, if the person is taking NMN, it's just converted to NR, and then, the NR is biosynthetically converted to NAD. So when we do quantitative targeted metabolomics, we can tell that the ratio of NAD to things like NMN and NR is like 100 to 1. It's all in the dinucleotide form. It's almost all in the dinucleotide form. What was I going to tell you one more thing, but I'm sure I'll come back to it at some point.
Yeah, we probably will. And so another thing, I think so you mentioned lots of different topics, and one thing I wanted to quiz you on is that I read how NAD plus fluctuates throughout the day due to circadian regulation. And so when you do these quantitative measurements and look at the levels in, I guess, mice that have been treated with a high-fat diet or not, does the variation, the difference between the groups, is that greater than the difference that you see in a circadian oscillation?
We were among the people that established the circadian oscillation with the late Paolo Sassoni-Corse, and for that reason, we always do our rodent sacrifices at the same time of day. Like right now, we're doing an experiment in which all the sacs are done at 4 p.m., which means that if we want to see a 1 hour effect of a supplement versus a 2 hour versus a 4 hour versus a 8 hour effect of that supplement, it means that since all of our sacs are done at 4, it means the 1 hour supplementation is done at 3 p.m., the 2 hour supplementation is being done at 2 p.m., the 4 hour supplementation is being done at noon, and the 8 hour supplementation is done at 8 a.m., full day for my my lab manager. And because we don't want to have that, we don't want to supplement all at the same time of day and then sac at different times, and then have that overlaid with the circadian, variation, which that would be not good. So I think in general, I can assure you that when we've done high fat and when we've done heart failure and neurodegenerative experiments, we've been controlled for time of day. And these disease models produce, profound effects that are much greater than the diurnal variation in the NAD system. Okay.
And I guess kind of on the flip side, there's also a term called chrono medicine, where actually the timing of supplementation is also important because the enzymes involved in metabolizing the supplements can vary throughout the day as well. And so do you think there could be time of day effects whereby there might be a more optimal time to take something like an NR supplement?
Yeah. There I guess 3 kinds of people in the world with respect to NR. Most people that I know get up and take it in the morning, roughly same time of day. There's some people that feel that it helps them sleep better, and so they take it at dinner or the same time, in the evening. And there are also clinical trials where they're going to doses as high as 2 grams a day, where they might be doing a gram twice a day. But same time of day kind of makes sense because it is a circadian function. Same time of day makes sense.
Okay, but no clinical trials so far have had everyone obviously take it the same day due to just the difficulties in...
There's not, there's not a specific clinical investigation of, AM versus PM effects. And, like the strongest effects of nicotinamide riboside that have been seen in the most number of studies are anti-inflammatory, so depressing the circulation of inflammatory cytokines like IL-10, IL-6, etc. That I think we showed that first with Gareth Lavery in Birmingham, UK, and it's been seen in a number of trials now. And so that would be the type of very predictable, outcome where you could see whether there's a slight difference in people taking it morning versus evening. But study hasn't been done.
Okay. And so you've mentioned as well a lot about inflammation and interferon response. And so I guess my background is cellular senescence, which one of its characteristics is the senescence secretory inflammatory phenotype. And there was a paper that I believe you're one of the co-authors of that came out either last year or the year before now, where it showed that the SASP, this inflammatory phenotype, I believe it increased CD38 expression on macrophages, and then that decreased NAD+. Yes. So would you be able to elaborate on that and explain what that's all about?
Yeah, it's with Anthony Covarrubias, who's now at UCLA. I'm at City of Hope, so we're at two LA institutions. And Anthony showed that there's macrophage subpopulations that have high CD38 and disturb the NAD system, and that's strongly a part of the inflammatory response. And I think that may be what's going on in a lot of things that you could term inflammaging. So if NAD is declining in the aging of a mouse in liver samples, one of the ways to explain that is that it's a higher population of inflammatory cells in the liver. It may be that the hepatocytes are identical, but the percent hepatocytes are what is altered, and that there's more inflammatory cells due to either transdifferentiation or infiltration or fibrotic processes, and those are going to be some really interesting things to figure out.
Yeah, for sure. And the other reason I brought it up is because I believe like glial cells are a form of immune cells, at least that's how they're derived. And so I think there's also a study that showed that CD38 increases in these glial cells in Alzheimer's. Is that right?
It is. In fact, I had a investigator in my lab on sabbatical with a strong interest in neuroinflammation that is continuing to work on that.
So given some of the studies we just brought up then, how would you go about describing what aging is and the role of NAD+?
What is aging? Okay, so I think aging is a developmental. I think aging is intrinsically linked to development, and I can defend that in multiple ways. First, the strongest, long-lived mutants that have been identified in animal systems as diverse as worms and rodents are loss of function alleles of genes involved in growth, right? So, DAF2 and insulin IGF pathway, and then the pituitary mutants in mice, including the Snell dwarf mice and the Ames dwarf mice and the pit mice. All of these genes that encode growth factor and growth hormone receptor and growth hormone releasing factor and so forth and so on. You knock out both copies and you get very small shrimpy mice that are infertile and very long-lived and very cancer resistant. And so you mutate growth, you alter the course of aging. I have a difficulty calling those longevity genes because the genes were actually conserved for growth and fertility. And in fact, in the development of animals, which is what, 750, 800 million years of evolution, animals evolved to be successfully born and to develop the ability for motility and acquiring our own food, and then recognizing mates and avoiding predation and reproducing, and either having lots of babies or protecting the smaller number of babies until they can successfully reproduce. And some animals can only reproduce one time, in which case there's really no selection for longevity. And some animals can reproduce multiple times, in which there's a slight selection for longevity, because let's say a fox that can live 6 years might have 6 times as many, broods, pups, or whatever foxes have, than one that can only reproduce one time. But is that really longevity, or is it fox cleverness that allowed it, and fox sex appeal and all the other things that allowed that fox to reproduce, over 6 years and, surviving 6 winters? It's, hunting ability. It's a lot of things. So I see longevity, the genetic components of longevity, as basically being emergent properties of selected traits. I don't really see lifespan as directly selected traits. There are no dominant longevity genes that have been identified in animals to my knowledge. I have a review article that's coming out on Friday, September 23rd. I'm not quite sure when this YouTube video will come out, but today is September 21. So in 2 days from today, I have a review article coming out with over 125 citations, the title of which is "Sirtuins Are Not Conserved Longevity Genes." I fully debunk the idea that sirtuins are dominantly acting conserved longevity genes from yeast to humans. It's just not true. The narrative that sirtuins are the primary mediators of NAD supplementation is not evidence-based. Connection between NAD and aging is that NAD is the linchpin of metabolism, and that our repair capacity and resiliency declines in aging, and many of those repair and resiliency reactions depend upon NAD and all of the accumulated insults involve, losses to the NAD system. So I think there is a use case for nicotinamide riboside in human health. It may be, in fact, more important as we age. There's lots of clinical trials involving NR to test these hypotheses. I'm evidence-based, so you don't see a supplement bottle behind me as I speak, and you don't see me getting out ahead of my skis and saying, making fanciful claims about extending human lifespan. But I'm excited about the clinical trials that are being done with NR, because what it does, in rodent systems is so powerful. And although we tend to like very striking quantitative phenotypes like protection from heart failure. So we tend to do shorter term experiments and then sacrifice the animals and then look at mechanisms, as opposed to doing 2 and 3 year long experiments and looking at, life and death things. But other people are doing those things as well. So I'm excited for the field, but I think that one of the problems is that it's deeply contaminated with some statements that are just not evidence-based at all. Like the idea that sirtuins are longevity genes is just not evidence-based at all. And so I think it's very problematic. And I spend a lot of time trying to, educate people on that.
Yeah, that's well said. And I guess you brought up quite a few points again. And one thing I just wanted to quickly ask you before I forget, so you mentioned about fertility, and have you seen in any of your studies any link between NAD plus levels and fertility? Because I know there's a lot in the news about the fertility crisis.
Yeah. We basically started, the connection with women's health and family health in an experiment that we published in 2019. It's Ear et al. And in Cell Reports. And you can see in the supplementary figure that we did an experiment with, that we did 2 initial experiments, and one didn't work very well and one was amazing. And so we went with amazing, and then we have a kind of a follow-up. But we wanted to look at the ability of nicotinamide riboside to, promote fertility in overweight and metabolically challenged black 6 mice. So they were on a high-fat diet, and then we tried to get them pregnant with and without NR, and there was basically only one, term pregnancy, and it was in high fat with NR and high fat without NR. Couldn't even get the mice pregnant. But you can't do a lot of analysis when you have one, litter of a supplemented mom and no litters of non-supplemented moms. And, it's just, so that's in a 2019 paper that NR might be helping mom get pregnant, particularly if she has metabolic challenges. But what happened in the normal chow side was really amazing. You think of mouse chow as being very nutritious stuff, and it has all the vitamins that it's supposed to have in it. It's been formulated. It has B3 equivalents in it, and yet the mice that were given additional nicotinamide riboside did much better. They had their during the course of their being co-housed with the pups, which is basically the nursing period, the mice were bigger and more resilient if the mom was NR supplemented, and the moms lost weight. And that we were able to attribute that to greater milk production, and it turned out to be linked to a better neurodevelopmental time course for the offspring. And not only did their motor skills advance better if mom was supplemented, but their whole life trajectory was better, and their body composition was better at 3 months, which is a very much of an adult rodent. And so they could be bigger at weaning, and then they could be leaner as adults. It was quite remarkable, and they had better, motor learning skills and Morris Water Maze performance, and they had lower anxiety scores, if you believe mouse anxiety scores. And so it was quite an amazing thing, and a lot of other people are doing things with NAD boosting in maternal health as well.
That's really interesting. And so you just looked at females, right? Or was there did you look at sperm count in males or anything? It was just females.
I don't think that our group has done sperm count. That could be interesting as well. Yeah, you can come and do a postdoc. I mean, reproductive aging is really, really interesting.
For sure, yeah.
And it's something there's very unique features of it in human, like the existence of menopause, right? So, half of a woman's lifetime is beyond her age of reproductive capability. That I think is because human babies are so inept at getting their own food and being able to be protected from predation, that in human evolution we needed not only mothers but grandmothers. But so there's some rapidly evolving genes that relate to aging, and then the difference between male and female is interesting as well.
Yeah, I was about to ask you, is there any sex-specific differences just in any of these NR studies that you've done?
Sex-specific
in terms of like efficacy, I guess.
Studies that we've done. Not so much that we've done. I have to say that our first high-fat thing was a male only. Isn't that terrible? And then we did this thing that was all female, but we did look at the pup scores, and there were male and female differences in a number of the figures. I can't cite you every single difference, but there were some differences. Yeah.
Okay, but this you think is something that we should probably pay more attention to, like going on.
We should.
Okay.
Absolutely. I sure am now. Yeah.
And so before, I think we've spoken in a lot of detail about your scientific research, but I thought because I'm a PhD student with both scientists, it'd be also interesting to hear your insights in terms of academia versus obviously you're an advisor, I believe, for Chromadex, which is this whether you have the NR formulation, and so how do you balance that as a researcher, and how do you find from my naive perspective, being a scientist, but also knowing you have ties with industry?
Sure, yeah. And for full disclosure, Chief Scientific Advisor, Chromadex, Chief Scientific Advisor of Juvenon, which is a Korean company that wants to make NR supplemented skin products, and a co-founder of Alfina, which is in the cancer metabolism space. I actually worked in industry for 5 years before grad school at Stanford, and the thing that I learned about it was teamwork. Someone very dear to me says teamwork makes the dream work, and that's something that you don't always see in academia. You have kind of small groups, fiefdoms, that sometimes compete against each other and don't communicate as much as they could. So it's great when you come up with something that could help human beings, that there's a way to license it and get folks to carry it through what's called the Valley of Death, and then see if they can make a minimal viable product from it, and see if it is something that could improve human health or animal health or something like that. So it's exciting when you can do it. And, if you have an opportunity as a PhD student to do kind of an externship in a commercial lab, you might learn some cool things. There's many different types of industry environments. I've been in 2 different ones, and in my consulting life, I've consulted for very different ones as well as big pharma. So it's a big world out there.
No, for sure. And I guess another question I have is, so there was recently a lot of news all over Twitter, and I think it was like a Nature commentary piece about the lack of postdocs, or a shortage of people applying for postdoc positions. And so obviously now I'm thinking about potential career choices. Where do you see academia going, and do you see there's going to be some shortages of researchers going forward if more are going industry, or where do you see like the future of academia?
I don't know if I can tell you what the future of academia is, Eleanor. I can tell you that funding is getting better in the U.S. And that there's a world of really good projects to work on. Pay is getting better for graduate students and postdocs in the U.S. I don't think it's a bad time to be a grad student or a postdoc in the U.S. I really don't. I think it's actually a good time to be.
Okay, well, that's good to hear then. And so some just final questions. What are some kind of big questions or interesting experiments you'd really like to see be conducted? You think either they're attainable but no one's done it, or things that you would like to see done, but
we're going to figure out where nicotinamide riboside is coming from. So nicotinamide riboside is a nucleoside like adenosine. There's such a thing as adenosine signaling. You know where adenosine comes from? It comes from ATP. So some cell is a NAD donor, basically. It's either a lytic process where the cell is blowing up and the NAD is being degraded to NR, or NAD is being exported for some reason and is being degraded to NR. But there's some type of cell communication that kind of creates the purpose of the nicotinamide riboside kinase pathway to salvage that NR and to be upregulated in conditions of metabolic stress. So that's something that you're going to see coming from us. You're going to see us publishing on a rare disease. I won't tell you which one quite yet, but we have a very exciting rare disease project that involves the NAD system. And we have a cancer story that's super cool. And we have that relates to DNA repair. And we're interested in obesity and inflammation, and we'll probably even do some repair type experiments that relate to problems that relate to human aging.
Awesome. And so before we just wrap up, do you have any final words of advice for my viewers who are either interested in going the scientific routes, any books or any sort of recommendations in that area, or finding advisors?
My biggest piece of advice is go through the actual data. So people are overly impressed with headlines and with people's interpretations of things that are incorrect. I think people should, distrust, received wisdom, look at the quality of the underlying data, and they're going to find a lot of really interesting things to work on. And I think it's very important to have a falsifiable hypothesis and to have go, no-go decisions like you have in industry. And so even though I'm in academia, we have falsifiable hypotheses and we have no-go, no-go decisions, and we prioritize our time and resources. And that's what you should do with yourself, because you will have only one life to give, and one Eleanor can contribute to science and the family and everything else that you want to do. So just be passionate and don't believe everything that you read, and you'll do great.
That sounds like good advice to me. So thank you for coming on today. It's been really insightful to talk to you. I can link, is Twitter the best place to direct my viewers to follow your work? Or
Sure. Yeah, I'm on Twitter at Charles M. Brenner. I answer questions every day, and it was a pleasure to meet you, and hope to interact with your audience over time. Take care.
Great. Thank you very much. So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.